Okay, chapter 12 is called How Genes Work. And really what we're talking about is how we're taking the information in our DNA and expressing that gene so that we can create the proteins that we need in our cells. So, the information contained in DNA is stored in blocks called genes on our chromosomes. The genes code for polypeptides, which will then become proteins. The proteins determine what a cell will be like, and we need them for every function we carry out. When we talk about the central dogma in gene expression, that just refers to this same process that all organisms use to express their genes. So, every single organism alive on Earth is using the same process of DNA, taking DNA, turning it into RNA, and turning that RNA into a protein. Those two separate processes are called transcription and translation. So whenever we're taking the information in our DNA and creating an RNA copy, that's transcription. Whenever we're taking that RNA and using it as the instructions to make a protein, that's translation. Also, whenever you think about what we're doing when we're transcribing, we're really just writing something down. So if you're trying to keep it straight which one is which, you could think of transcription as writing down the information in DNA as RNA, and then translation as translating that information into a language that we can use, the protein. So we're summarizing the two steps. First, we have transcription, which is where our genetic information passes from the genes to an RNA copy. That's like writing it down as RNA. And then our RNA copy will direct the assembly of a chain of amino acids, which are going to form a polypeptide, which will then become a protein. And that process is translation. Transcription, we're turning DNA into RNA as a copy. Translation, we are taking that RNA copy and the instructions that it carries to create the protein. Whenever we're making RNA, there are really three main types that we could be creating. So the first is messenger RNA, and that's the actual information that is the copy from our DNA molecules. So you can see that it really resembles a single strand of DNA. You can see that each of these is representing a nucleotide along the path. This is going to carry the information for the specific protein we want to make. Next, we have our ribosomal RNA. This is part of the ribosome responsible for recognizing and binding to that incoming messenger RNA. So the messenger RNA is going to come out of the nucleus, go find a ribosome, and then that ribosomal RNA that's part of the ribosome is what's going to allow for the messenger RNA to know where to initiate. We also have transfer RNA. And these RNA molecules are responsible for bringing the amino acids that are going to become the polypeptide chain that eventually becomes the protein. If you'll notice, we've got 1, 2, 3 nucleotides on the bottom of this tRNA molecule, and we're going to get more into that later on. But I just wanted to point out that that's happening. Transfer molecules have 3 nucleotides that are going to line up with our messenger RNA later on. So when we're talking about transcription and we're making RNA from an RNA copy from DNA, we could either be making mRNA, rRNA, or tRNA. This is messenger, ribosomal, and transfer. In translation, we are taking the information in our messenger RNA to create a protein. This whole process is really gene expression. So we have genes. How do we express them to actually create what they're intended to create? That's this whole process of transcription and translation. Both of those are part of gene expression. So I mentioned that transcription happens in the nucleus. That makes sense because we know all of our DNA is housed in the nucleus. We're not going to bring it out where it's going to be at risk of getting degraded by different enzymes or broken down or damaged. We leave it in the nucleus. It stays there. So that makes sense that our transcription is happening inside the nucleus where we're making our RNA copy of one of these strands of DNA. Also, it doesn't really matter which side of this DNA double helix we're copying because the information is the same. Even though this may be an adenine while this is a thymine, the information is still going to match in the order when we make a copy because the opposite is still going to be the same order of nucleotides. Whenever we've made our RNA copy and it's ready to leave the nucleus, it's going to leave through the nuclear pore, and then it's going to go find a ribosome so that it can make that protein. You might remember that we have a bunch of ribosomes on our rough endoplasmic reticulum. We also have a lot of free-floating ribosomes scattered throughout the cell as well. Our DNA stores genetic information safely in the nucleus, and that's why that's where we're making our RNA copy. Messenger RNA is that RNA copy of the gene that is used to produce the polypeptide. So this is the one with the actual instructions. A protein called RNA polymerase is the one that produces the mRNA copy, the messenger RNA copy of DNA during transcription. And that should be pretty easy to remember because if you remember in our last lecture we talked about DNA replication, and we know that DNA polymerase is the enzyme that brings all of the new DNA nucleotides in when we're making a copy. Well, in this case, we're not making a DNA copy, we're transcribing. So we're making an RNA copy, which means we use RNA polymerase as the enzyme that is bringing in the new RNA nucleotides to make this copy. You might remember that DNA polymerase had to show up and find a spot called the primer, which is like the spot where it could start and kind of kick off the DNA replication. So DNA polymerase is looking for a primer. RNA polymerase is the enzyme that's looking for a promoter. So you could think of it in the same way, but they do have separate names. It's the spot that RNA polymerase is going to start making that copy. So RNA polymerase is going to assemble the complementary copy, again in the 5' to 3' direction, adding nucleotides to the 3' end. RNA polymerase is going to pair guanine with cytosine, as always, but we're going to have a change because adenine, instead of being paired with thymine, is going to have to be paired with uracil. That's just a thing about RNA. RNA has a slight change to one of the nucleotides, which means that adenine is going to now bind with uracil instead of thymine. It really is pretty simple to remember and figure out any problem that you need based on this difference, because you know guanine binds with cytosine like cars go in garages, you know adenine binds with thymine like apples grow in trees, and if you just know A binds to T, but when we make RNA we're replacing T with U, that's a pretty simple path forward to be able to figure out any kind of nucleotide arrangement that you need to. It's just one substitution. So in transcription, again, we have to have an enzyme come in and open up our DNA double helix, and that's what's happening here. It's that same helicase enzyme. And once the helicase enzyme has opened up our DNA strands, you can see we have to have a spot to initiate our RNA copy, and that is the promoter. So the promoter is the spot RNA polymerase is going to show up to start adding nucleotides. You can see here that on DNA, this adenine was paired with a thymine, but since we're making an RNA copy, now it's uracil. Cytosine is still pairing with guanine. Guanine is still pairing with cytosine. This adenine was paired with thymine, but now it's paired with uracil. The thymine still exists on our DNA strand. It will pair with adenine on our RNA strand, and so on. That's going to continue until we reach the end of our coding segment. And then our mRNA strand is going to exit the nucleus. To correctly read a gene, a cell first has to translate the information encoded in the DNA into the language of proteins. And all of that translation is following the rules called the genetic code. So the messenger RNA is read in 3 nucleotide units at a time. You could almost think of them as the words, but they're 3 nucleotides and they're called codons. Each codon is going to correspond to a particular amino acid. When we talk about the genetic code, it was determined from trial and error experiments to work out which codons matched with which amino acids. And that was a long, involved process with a lot of different scientists. But we also found that the genetic code is actually universal. It's employed by all living things. So dolphins, mosquitoes, black bears, humans, chimpanzees, anteaters, manta rays, all of us are using bacteria. All of us are using the same genetic code. And here it is. So don't let this table overwhelm you. It's much simpler than it looks, but it really is amazing to think that all of these nucleotide combinations resulting in these 3 nucleotide codons are responsible for all of life on Earth. Like, every combination is creating every amino acid that's going into a polypeptide chain to make a protein that we need for all the different things that we do in our cells and in our lives. So to help you simplify this when you're looking at it, we know that codons are made up of 3 letters, and this table is a really convenient way to break that down. So for instance, if we had a codon of UCA, we would look at first this side to look for the U letter. Next, we go to the second letter, C, so that lines up here. And then here is A, our 3rd letter, and that lands us here. UCA must be coding for the amino acid serine. If we could do that, another example, let's do GUC. So first letter G, second letter U, 3rd letter C, we are coding for valine. That's the amino acid. It's also important to note that we have 4 codons that have that are not specifically for coding for an amino acid. They're to indicate the beginning and the end of the stretch that's being copied or that's being translated. So we've got AUG, which codes for methionine and is also our start codon. So that is the beginning of any translation. We'll have AUG. Once we're at the end of the code of the gene that we're trying to express, we will either reach a stop codon that says UAA, uracil adenine adenine, uracil adenine guanine, or uracil guanine adenine. All of those are stop codons. So once we get to that point, there's not going to be an amino acid associated with those codons. So that's how we know to stop our gene expression, to stop making our polypeptide chain. We know that translation is occurring on ribosomes because we've talked so many times about ribosomes being the protein-making factories of the cell. We know we're making proteins in the ribosomes. As you guys might remember, we've got a bunch of ribosomes on the rough ER. We've got a bunch that are floating freely in the cell. Each one of those ribosomes is going to be a complex of proteins and also several segments of ribosomal RNA, which is basically telling us where the mRNA is going to line up. Our ribosomes are comprised of 2 subunits. We've got a small subunit and a large subunit. The small subunit has a short sequence of its rRNA exposed that is identical to the leader sequence that begins all genes. So mRNA is going to bind to that small subunit. And here we can see what that looks like. So here's our ribosome. We've got our small subunit, our large subunit, and then this is what's going on inside. So mRNA is going to enter and be fed through the ribosome. tRNA is going to bring our individual amino acids to this A site, which is the first of our 3 sites. We've got an A site, P site, and E site. And our transfer RNA is going to line up with the codons on our mRNA and bring an amino acid. Once we get to the P site, those amino acids are going to be able to create peptide bonds. And then once that transfer RNA has released the amino acid, it will be able to exit through the E site. So if you're trying to remember the order of things, try to remember that A could be like arrival, we're arriving at the ribosome, and E is like exit. Then you could also consider P for peptide bonds. That's not what they stand for, but that's just to help you remember their purpose. So the large RNA subunit has those 3 binding sites, and those binding sites are for the transfer RNA molecules. These binding sites are A, P, and E, and it's the tRNA molecules that bring the amino acids to the ribosome. The structure of a tRNA molecule is important for its function. So it basically holds the amino acid attachment site at one end, and then it has a 3 nucleotide sequence at the other end, which is corresponding to the specific amino acid it's carrying. So this 3 nucleotide sequence is called the anticodon, and it's complementary to one of those 64 codons in the genetic code. So basically our mRNA has codons, which are 3 nucleotides, and then our tRNA has the complementary anticodon, which are going to pair up with those 3 codons on mRNA. We also have activating enzymes, which match the amino acids with their proper tRNAs. So here you can see what that looks like. This is an example of tRNA. And here is the bottom with the anticodon. This is a combination of 3 nucleotides. At the top, that's where we attach the amino acid that's going to be part of our polypeptide chain. So it's important to remember mRNA is our codon, matching with tRNA that has the anticodon. Once an mRNA molecule has bound to that small ribosomal subunit, the other larger ribosomal subunit is going to bind as well. And that's what's going to form our complete ribosome complex. During translation, the mRNA is going to thread through the ribosome 3 nucleotides at a time. So you can see it going this way. Our tRNAs are coming in this way. So they're going opposite directions. So a new tRNA that is holding an amino acid is going to be added and enter the ribosome at the A site. So tRNA binds the anticodon, these 3 nucleotides, to the matching codon, these 3 nucleotides. At that point, it will move to the P site, where the amino acid gets to line up with the other growing chain of our polypeptide, and our peptide bond can form. So now this amino acid will be attached to our polypeptide. Once that amino acid is released from tRNA in the P site, we're going to go to the E site, and that's where our tRNA is going to exit the ribosome. You can see it still has the same anticodon. It's going to go pick up the same kind of amino acid as the one that it dropped off, and it'll be recycled back into this process and come back and find another ribosome with another gene to translate. Before a new tRNA can be added, that previous tRNA in the A site is going to have to shift. So that makes sense. We have to make room. It's kind of like a factory line. So the tRNA moves from the A site to the P site, so the amino acid can create that peptide bond with the growing chain. And then now the empty tRNA in the P site will have to shift to the E site, where it can exit the ribosome and be released. We can see that same process happening here, but it's kind of helpful to see it with the amino acid names and with the actual nucleotide arrangements shown for our codons and anticodons. So again, our mRNA is here. The anticodon on our tRNA is going to align with the codon on our mRNA. You can see here we've got cytosine pairing with guanine, uracil that used to be thymine pairing with adenine, and guanine pairing with cytosine, just like we would expect. C with G, A with T, T is replaced by U, uracil. So we get that alignment. That means that the A site, the tRNA and mRNA are adjoined. We go to the P site so that a peptide bond can be formed between our amino acids. So here they are forming that peptide bond between our amino acids. Once that amino acid is released, we've got our empty tRNA. So this is going to go pick up another of the same amino acids that's going to be needed in the next protein synthesis. So that process just continues until we've made our actual polypeptide chain that polypeptide chain is going to go off to another organelle to be folded the right way, to be in the right form to actually be a protein. Because right now we have the beginning of our protein with this polypeptide chain. We've got the right amino acid arrangement, we just don't have the right shape yet. So that's the next step. Whenever we reach our stop codon that we mentioned before when we were looking at our genetic code table of all of our amino acids, the stop codon is what signals the end of that protein. So once we've got a tRNA with a stop codon in, once we've got an mRNA with a stop codon, we're not going to have any more amino acids that are added to the chain. And at that point, our ribosome complex will fall apart and our newly made protein is going to be released into the cell. So here you can see a side-by-side of the processes we've talked about. So we've got DNA replication, which we talked about in chapter 11. Let's just kind of go over the differences of these processes really quickly. It's nice to have a side-by-side to compare them and keep the different steps separate. So in DNA replication, we're not making an RNA copy, we're making an exact DNA copy. And for that, we talked about how we have helicase that shows up and unravels our DNA double helix so that we can open it up and access those exposed nucleotides to make our copies. So helicase opens up the DNA double helix, then we're going to have a leading strand of DNA copy. The DNA polymerase, which is the enzyme that brings new nucleotides, is going to look for the primer, which is the spot that it's going to start adding nucleotides. So the DNA polymerase goes to the primer. After that, it adds nucleotides in this direction towards the opening of the double helix. So that makes this the leading strand because it is uninterrupted. It's going towards the unzipping motion to add nucleotides, and it can just go one after another until that copy is complete. This side is more problematic because we can only add to the 3' end of a strand, and this action is going to be going away from the unzipping of our DNA. So as a result, we end up with a lagging strand that is kind of more complicated and slower. It's not as efficient as the leading strand, but we'll still end up with an identical copy. In this strand, we're going to replace a segment of about, well, really the number is random, but you could just imagine, for example, that we're placing 5 nucleotides, then going back, placing 5 more, then going back, placing 5 more, and so on. So we call those specific fragments that we're placing together Okazaki fragments, named after the scientists that discovered them. Once we've placed all of our nucleotides on our leading and our lagging strand, we're going to have the enzyme ligase come in and seal those nucleotides in place. Once that happens, we have copied our DNA, and we know that this is a semi-conservative replication because we have somewhat conserved the original DNA in both this copy and this copy. You can see there's an original strand of DNA here and a new strand of DNA in red, and this one has an original strand of DNA here and a new complementary strand in red. So that's replication. Now transcription is when we're making an RNA copy of that DNA information. And then we're using RNA polymerase instead of DNA polymerase, which makes sense because we're putting RNA nucleotides in instead of DNA nucleotides. So once again, we're going to have an enzyme open up our DNA. It's still going to be helicase. We will have a spot to start our copy. Instead of that being the primer, like it was up here for replication, we call it the promoter. And that promoter is where RNA polymerase knows to start adding RNA nucleotides. So we're adding our nucleotides until we reach the end of the gene to be transcribed. At that point, our transcription is complete. That RNA might be edited a little bit before it leaves the nucleus. then the RNA is going to go out into the nucleus and find a ribosome so that we could enter translation. Now here's translation. You can see we've got our mRNA and our ribosome. You can see the mRNA has all of these codons that align with the anticodons on tRNA molecules. That's going to determine which amino acid the tRNA brings in. And once they come into the A site, they will move to the P site so they can form peptide bonds to continue this polypeptide chain. Once that bond has been made, the empty tRNA molecule will be released to go find another amino acid matching the anticodon sequence that it has. And as a result, we'll end up with our polypeptide. So the prokaryotic gene is an uninterrupted stretch of DNA nucleotides, which we know they have that circle of DNA. In eukaryotes, the coding portions of the DNA nucleotide are going to be interrupted by non-coding sections of DNA. So that's interesting. We haven't really talked about this yet, but it's not like every single nucleotide in our genome is going to code for a protein to be produced. A lot of those sections are not coding for anything at all. Some of them have a purpose. Some of them have a purpose. Some of them we have not discovered a purpose for. A lot of the times that purpose is structural. It's suspected that a lot of these instances also there is a separation between important genes. So there's decreased likelihood that we're going to have a mutation or an error in transcription where we're combining the information from those genes because there's this long section that's not coding for anything in the middle, separating them. Those sections of our DNA that do not code for proteins are called introns, and the sections of our genes that do code for proteins are called exons. If you need a trick to help you remember which is which, you could think about the fact that when we're expressing our genes for gene expression, that's an exon, so expression, EX, that's the one that's being expressed. That may help you differentiate, but do try to remember introns are the non-coding portions of our DNA, exons are the coding portions. Also, when a eukaryotic cell first transcribes a gene, it's going to produce what we call a primary RNA transcript of that entire gene, which is going to include the introns and the exons, the part we don't need to code and the part we do need to code. That primary transcript is processed in the nucleus before the mRNA leaves. So that means we're going to take out, basically cut out the introns from our RNA and seal it back together so that we have only the genes that are coding for proteins in our copy RNA strand. Also, before it leaves, we're going to add a 5' cap and a 3' poly A tail that are there to protect the RNA transcript from degradation. So you could imagine this is what we have at the end of transcription. This is our DNA. This is what we have at the end of transcription. We have included all of the information from our coding regions, the exons, and non-coding regions. We're going to cut out the introns because we don't need them for our protein expression to create our proteins. So we cut those out and splice together our exons, along with adding a 5' cap and a 3' poly A tail. These are added to keep enzymes from degrading our mRNA strand before it can reach the ribosome and create our polypeptides. So you could think of it like an end that keeps this from being broken down in our cells. If it were floating around in the cytoplasm looking for a ribosome and it didn't have this cap and this tail, it could just be degraded before it even gets to a ribosome to be used. So in humans, exons can actually be spliced together in different ways. And using these different combinations of the exact same exons can produce different proteins. And that term, the term for that is called alternative splicing, meaning at different parts we can splice our different exons together. So we have about 25,000 genes in the human genome that appear to encode as many as 120,000 different mRNAs. So we have so many more messenger RNA possibilities than we do have individual genes in our DNA. The different arrangement of those exons is changing the proteins we're making. So you could consider it like this. We cut out all of the introns, we stitch all the exons together, but you could also imagine in another instance, maybe this exon was placed here in the middle and this exon was placed here at the beginning. Now we have a completely different protein, even though we're using those same exons of the gene. And that's alternative splicing. So when you're trying to think of what accounts for the reason that we have so many different mRNAs to code for so many different proteins than the number of genes we have in our human genome, it's because we can splice them together at different points to create different proteins. Also, in prokaryotes, once again, they're so simple. A gene can be translated as it's transcribed. So it's kind of a one and done process in that circular ring of DNA. In eukaryotes, we have that nuclear membrane that's separating the processes of transcription, where we're copying the DNA into RNA, and translation out in the cytoplasm on a ribosome, where we're making that protein. And that makes us much more complicated in our process. Here you can see just an example of that kind of one and done transcription and translation in prokaryotes. We've got RNA polymerase and ribosomes making our RNA copies of that DNA information. This is a really great slide to break everything that I've gone over down further. I'm going to go through it really quickly, but I do think that this repetition will help all this information stick for you because it is kind of a highly specific process. So in the cell nucleus, we've got our RNA polymerase. It's going to look for the promoter on our DNA strand to know when to start transcribing a specific gene into RNA. We're going to start that process and keep going until we've got our complete mRNA copy. At that point, we're going to cut out the exons. So we're going to cut out the introns and get rid of them. We're going to keep the exons and stitch them together using the enzyme ligase to seal them together. We'll also add that 3' and 5' cap and tail. That's going to keep the mRNA safe until it finds a ribosome so it doesn't get broken down. Once it finds a ribosome and attaches to some ribosomal RNA, then our large ribosomal unit's going to come together with the small ribosomal unit so that we have our initiation complex. Here we have our small and large units of our ribosome. We've got mRNA here moving through the ribosome in this direction. tRNA is bringing the anticodon that matches the codon on mRNA so that we can line up our amino acids. They arrive at the A site, go to the P site to create their peptide bond, and then exit the ribosome through the E site so that they can go pick up another amino acid and come back in to the same process for a different protein. And then our amino acid chain is going to become our polypeptide chain. It will be released and go off to other organelles to be sorted and folded into the right protein shape for the right function. So every cell has to be able to regulate when particular genes are used. And prokaryotes control transcription in various ways. They are controlling gene expression by saying when individual genes are to be transcribed. So when should this be allowed? In our prokaryotes, genes can be turned off whenever a repressor binds to a site called the operator on the DNA. And that is going to block access to the promoter. So we know that the promoter is the spot we have to show up to start RNA polymerase bringing those RNA nucleotides. But if a repressor protein binds to the operator, that's going to block the promoter completely. So now we can't start adding any RNA nucleotides to make our copy. Also, genes can be turned on by an activator, which is a protein that makes the promoter kind of more accessible to RNA polymerase. You could almost imagine it like increasing the magnetism between RNA polymerase and the gene that needs to be copied. Whenever we're talking about a segment of DNA that has a cluster of genes that are always transcribed together, that's an operon. So you could think of it like a group that's going to stick together. They're like, no, she's not going without me. He's not going without me. We all stick together always. These genes will always be transcribed together. One example of those genes is the lac operon in the bacterium Escherichia coli, which is also known as E. coli. This operon contains genes that code for enzymes associated with breaking down lactose. And the operon contains regulatory elements, the operator and promoter. And the presence of that lactose is going to affect a repressor protein and cause it to fall off the operator, which is going to make it possible for RNA polymerase to then transcribe the operon, which is the group that's being transcribed together. Whenever we're talking about gene regulation in eukaryotes, it's geared towards the entire organism, not just the individual cell's needs. So the chromatin structure of eukaryotic chromosomes can be altered in several ways that are going to affect transcription. In chromatin, DNA is packaged around those histone proteins, and we know that once we've got a unit of 8 histone proteins with chromatin wrapped around it, that's called a nucleosome. Well, if we modify the histones, we can actually end up with a greater condensation of the chromatin, which can make our promoters less accessible for gene transcription. So basically, we slightly alter the histones. That's going to create more condensation, so like kind of a tighter wrap of our chromatin around the histones, which is going to make it harder to get to the promoters, which is going to make gene transcription less possible. So that's a way to slow down transcription when we don't need to make protein copies. There's also the option of methylation of the DNA, which can also ensure that any kind of turned off genes are going to stay off. It's basically adding a carbon-hydrogen group that makes the affinity for adding new nucleotides to where we're not going to do that. So our turned off genes are no longer going to be able to code for proteins if they've been methylated. Here's an example that's showing our histones. Once we've got a complex of 8 of them with chromatin wrapped around it, that's when we call it a nucleosome. Our eukaryotic transcription also requires proteins called transcription factors. And these will form an initiation complex, and they are also part of recruiting RNA polymerase to the promoter. So they're helping. You could think of them kind of like road signs along the way, pointing to the promoter, so the RNA polymerase knows where to go. Eukaryotic genes also have sequences called enhancers, and that's what our activators will bind to. So often we have enhancer sites that are located far away from the genes that they actually influence. And then the DNA can bend to bring the activator in contact with the RNA polymerase initiation complex. So the activator is really just acting to make our RNA copy during transcription faster. It speeds up the process. It makes it happen more efficiently. So the activator binds to the enhancer sequence. You can see our transcription factor here is binding at the beginning of the promoter. We will bend our DNA strand so that our activator and transcription factor can align with RNA polymerase and kick off the transcribing of our coding region of the gene that comes after the promoter. So here you can see another example where we've got activators binding to enhancers. This can become more and more complex in shape, but our enhancers and activators are interacting with RNA polymerase at the promoter region in this initiation complex. And once all of these connections have been made, we're going to have a more efficient process transcribing the DNA into RNA by RNA polymerase. Also, let's go back for a second. You can see here on this image, they call them basal factors. They are still just transcription factors, so don't let that confuse you. Our RNA molecules can also play a role in regulating gene expression. So we have something called small interfering RNAs, which can block the transcription of genes whose sequence is complementary to the small interfering RNAs. So if we look at the name, small interfering, these RNAs are small and they're in the way of gene expression. These are mechanisms of RNA interference, which can also be referred to as a mechanism of gene silencing. So basically we have a gene that we want to copy, but these guys are stopping that from happening. Small interfering RNAs. And they're doing that with the same kind of seats taken approach that we've seen before. Basically, our small interfering RNAs are going to attach with the mRNA copy that we've made, and they're going to have a matching nucleotide sequence so that they attach all the way along the length of that mRNA copy. And that means now mRNA, all of the codons on mRNA don't have any spot open to bind tRNA. So that means there's nothing that they can do. They can't be translated. That bond is already made. There's no open seat for tRNA to bring the anticodon to. And as a result, they will be destroyed. So the small interfering RNAs will bind to them and then they will be destroyed because they're no longer useful. And that is a mechanism of gene silencing. When we talk about eukaryotic gene expression, it is controlled at many stages. Control of our gene expression can be affected by all of these things. So it can be affected by our chromatin structure, including how it's wrapped around histones and how condensed our chromatin is. It can also depend on the availability of our transcription factors. So we don't have an unlimited number of transcription factors. So that's going to depend on what proteins are already being made and what transcription factors are available. We also have alternative splicing, which will affect our gene expression. That will determine what order the exons are placed in whenever we have spliced out our introns. When we talked about RNA interference and gene silencing, that was our small interfering RNAs that are able to basically throw away an mRNA copy that's no longer needed or that should not be used to translate into a protein. Also, just the availability of our translational proteins. We don't have an unlimited number of those either, so that's going to affect how fast and how often our genes are transcribed and translated. And then there's also the possibility for chemical modification of a protein after it's been made. So it's not like, okay, we strung all the amino acids together, this is a completely finished product. There could still be chemical modifications that take place after that. Now we have covered chapter 12. This is a useful slide if you want to go over any of the things that we've talked about. It's kind of a nice summary and overall view of things as they're happening through transcription and translation. The main video summarizing this process that I would like for you guys to watch once, if not twice or 3 times, is posted on Canvas. And it would be really great if you could go over that video and make notes as you go, because it's going to make such a difference for you if you could watch the video and every time they say the next step, you write it down with your own handwriting. That really does help you remember a detailed process for a really long time in a way that other things just don't. So I highly recommend taking notes as you go when you watch that video, but do remember your lecture is not complete until you've gone to Canvas and watched that summary of transcription and translation, because it really helps to be able to see the visual from beginning to end. Thanks, guys. That's it for Chapter 12.